So our next talk is going to be from Kamel El Omari. So Kamel is a senior beamline scientist at the long wavelength beamline, so I-23 at Diamond Light Source. And he's also a lecturer in biochemistry at the University of Oxford. His main research interests are in viral host interactions and sulfur SAD phasing. And today he's going to tell us how we can find and locate not just sulfur, but lots of light atoms through anomalous scattering.
Okay. Thank you for the introduction. So this session is about model building, and I'm hoping to show you that actually you can use anomalous data to help you with your building or get better get better structure and be more confident about the data that you deposit. To follow up on Dorothy's talk, we had two evolutions in structural biology, the cryo-EM about 10 years ago, and now the AlphaFold 2 or Rosetta Fold revolution. It has changed the way we work, even just from designing your construct for cloning. I come from a beamline where we used to do a lot of experimental phasing, and clearly this has dropped a lot. AlphaFold can also help you to build your model or predict protein-protein interaction. The issue is that it's so good that some people think that it's good enough to not use experimental data. And we are lucky to have like papers like this one that Dorothy mentioned in our talk that really put back things in context and tell us that we do need experimental data. We do need it for dynamics, for high resolution, for X kind of type of experiment. To solve nucleic acid structure, these are quite difficult still actually. And finally, for element identification. And I'm going to talk about this. So I will start with an example that of AlphaFold success. So when AlphaFold came out, we had this data that was collected 20 years ago, so before I started my PhD, and that has never been solved. There were a couple of similar proteins in the PDB, but it wasn't just good enough. And so we had to unarchive the tape and get the data. And actually, AlphaFold worked straight away. And it was almost quite scary how good it was. Now, if I display here the 5 AlphaFold models, you see that they are quite similar to the final model at the end in gold. And the core is actually similar to the other structures in the PDB. But the different part that is in red is actually the important part, at least biologically important for this structure, at least for us. And you can see that it wasn't predicted properly like here. A bit coming back to what Dorothy has said, like you shouldn't, AlphaFold is extremely good, but you shouldn't have a 100% confidence into your model. You do need experimental data. Going a bit more into this, looking at the electron density map, we see a nice density here, and that's a polymerized. So we know that you would expect some magnesium or divalent cation to be bound. The question is, what is there actually? So it's known that magnesium is necessary for the enzyme, but if you look at similar enzymes, they also need manganese. They can bind manganese, and they can also bind calcium. So which one of these should we model there? Now, obviously, we have looked at the crystallization condition, and it's full of calcium. So it's likely to be calcium. But if you're going to make a hypothesis about the role of the calcium, you need to have a bit more confidence into the fact that it is a calcium. You need a different way to justify that it's a calcium, apart from the fact that it's present in the condition. So, as I said, it was collected 20 years ago, but at least we are lucky that it was 360 degrees of data. It was actually a high symmetry as well. That means that we could use the anomalous signal present in the data set, even if it's weak. And we did use that for F-double prime refinement. And I will come later to F-double prime. So, but just to show you the results, the data was collected at 0.9 angstrom wavelength. The theoretical value for F-double prime for calcium is 0.5, magnesium 0.06, and manganese 1.2. We did the refinement in Phoenix. We refined F-double prime, and it came quite clearly that it's very close to a calcium. and it can be a magnesium, and manganese is about twice the value. So this gives us more confidence that it is calcium. And this is based on your data. It's experimental. Now the issue is that you need to have you need to do that at the end of your refinement. You need to make sure that your model is quite right to use that. But we have better way to do that by collecting the data a bit differently. And I'll come back to that after. So in conclusion for this, as Dorothy said just before, AlphaFold is great for molecular placement. And without AlphaFold, this project wouldn't move forward. But you still need experimental data to make sure that your model is right, and also in this case to identify the metal there. And as I say, we can actually do better than f-double prime refinement, but if it's the only thing that you have, it's already quite good. So since it's a study weekend, I would just give a quick recap on what is anomalous scattering. That's mainly for students. So in normal scattering conditions, you consider that your electrons are free. They're not bound to the nucleus. And when the X-ray beams interact with electrons, they are going to oscillate with the X-ray beam. And by doing that, if you basically oscillate a charged particle, it will itself emit X-rays. In anomalous scattering condition, it's a bit more complicated because now we assume that the electrons are bound to the nucleus. For example, the inner core electrons in the K shell have an energy of binding of 3.6 keV, roughly. So if you use X-rays that have the energy similar to the binding energy of 3.6 keV in this case, you will have an absorption phenomenon. And that's why we call this the absorption edge. And a few things can happen there, but one of the things is that the electrons will absorb the energy and can be ejected from the nucleus, or it will also, you will have a phase shift in the scattering of the atom. So that means that the atomic scattering factor needs to be corrected. If we call F0 the normal scattering factor, that is independent from the energy of the wavelength, we correct it with F prime, which we call also the dispersive differences, which is always 180 degrees out of phase. And with F double prime, which is always plus 90 degrees out of phase. And this F double prime is the F double prime that we refined just before. Now, F prime on F double prime, so you will get your new corrected atomic scattering factors. And this is not up to scale because F prime and F double prime are pretty small usually compared to F0. F prime and F double prime depends on the wavelength. So here we can display their theoretical values. Either you go on the Washington website here, or you use pulse bond app that you can use on your phone to show you exactly the same thing. So what it shows here is that actually you always have a value of F prime and F double prime that are kind of different from zero. So you can actually consider that you are always in anomalous condition. What we call normal scattering condition is when these values are so close to zero that we think that they are negligible. The F double prime is maximum here, and that corresponds to the absorption edge, and that's the energy that I mentioned before that will trigger a transition in the electronic shell. So if you want to measure some SAT data, for example, here for potassium, you will basically you choose a wavelength which is right here at the right above the peak. Another consequence of anomalous scattering is that it will change the Friedel, it will affect the Friedel's law. So if we look in red here at the structure factor of your protein, or the sum of all the atomic structure of all the atoms in your protein, and we add an anomalous scatterer, we add F0 that is independent from the wavelength. We are going in anomalous scattering condition on the right to correct it with F prime on F double prime. Okay, and because as I said before, F double prime is always 90 degree, plus 90 degree, we are losing the symmetry here. So when you calculate the total scattering structural factors of protein plus anomalous scatterer, now you see that these two reflection, HKL on minus H minus K minus L, they don't have anymore the same amplitudes compared to normal scattering condition. And these differences is what we use either for phasing or for or to pick up the anomalous sites. Now, we had this morning a talk from Elsbeth about the radiation damage. These differences that you see here are actually very, very small. They are just a few % of your amplitudes of your intensities. So this will basically disappear straight away if you overexpose your data. So radiation damage is very bad for anomalous signal. So you need to be very careful if you collect anomalous data. Okay, now you can use anomalous data for experimental phasing. And as it was said just before, the use of experimental phasing has decreased a lot. So when AlphaFold came, since I'm working on a phasing beamline, we had a few structures solved by native SAT, so structure that couldn't be solved by molecular placement with deposited PDBs. They are very different, they are very diverse structures. And we had at that time around 18. And one of the placement students tried to see if basically this structure could be solved with AlphaFold models. And actually over the 18, only one couldn't be solved. They were more or less easy to solve, but at least 17 of the 18 could be solved. So it's clear, and we see that even now, that experimental phasing has largely decreased, or the need of experimental phasing. But there are cases, and it was mentioned just before also, where especially with nucleic acid, and I'm speaking about nucleic acid not bound to protein, just nucleic acid, they are very tricky to solve by molecular placement. And for phasing, you need to either use heavy metals or modify your nucleotides. And this is not always possible, actually. So we did solve a few with naturally binding ions. Like, for example, this one was an RNA quadruplex bound to potassium. So we use the potassium to solve it. But these are quite difficult. And I would like to mention also that many people come to us asking to do phosphorus SAT. You've got many phosphorus 1 per nucleotide in this type of structure, so a very strong signal. But actually, it's extremely difficult to do. And the reason for that is that it's not a question of anomalous strength. It's a question of number of unique reflection. Tom Twalinger published a nice paper saying that basically, to be successful with SAD, you need to have low B factors, a high number of unique reflection, and a low number of anomalous scatterers. And because these structures are usually quite small, the unit cell is small, so the number of unique reflection is small. So these are, unless you've got resolution close to atomic resolution, it's difficult to do. So hopefully the next algorithm of machine learning will help us with this issue. So now coming to element identification. So you can identify elements like, for example, zinc or iron in standard beamlines. So I worked on the long wavelength beamlines at Diamond, I-23. And the advantage of I-23 is that we can basically access the absorption edges of all these elements: phosphorus, sulfur, chlorine, potassium, calcium. And these are quite important elements in biology. You do find them in cells, in your crystallization conditions. They are used by your protein or your enzyme for their functions. So the work I'm going to show you now, everything has been collected on I-23. So how do we identify elements? We start usually by doing a fluorescence spectrum, and I should say that everyone should do that, even if you are working on standard beamline. That will at least tell you what you've got in your crystal or in your condition, because sometimes your protein picks up ions from your expression system that you're not even aware of. So the fluorescence spectrum are quick. They are so quick that there's almost no damage. and gives you an idea of what you got there. Then we choose the absorption edge or the element, and we collect two data sets, one above and one below the absorption edge. I should say we collect them usually in the with the interleave method. So we basically, for example, we'll collect 20 degrees above the edge, then 20 below, then 20 above and below. We alternate, and this is to spread the dose. As I said before, radiation damage can be very bad for anomalous scatterings, for anomalous data. So we want to spread the dose to be able to compare these two data sets. Then the next step would be to calculate phase anomalous Fourier difference maps. If you don't know how to do that, as Graham said before, please do ask your beamline scientist. And if you don't want to do it, actually it's done automatically for you in iSpy-B. So but you do need to either give a sequence for an AlphaFold model to be created, or give a PDB. This is for Dimple to be able to run your molecular placement. Then anode is used to calculate your difference map. So you will actually find a code session in the Dimple folder where you can just display your anomalous Fourier difference map. So really there's nothing to do apart from supplying a PDB or a sequence. So let's say, as an example, like if you want to identify chloride in lysozyme. So this is, if you look here at F double prime for lysozyme, we are going to collect here above the edge and here below the edge. The edge is 2.8 or PVKH. Because it's lysozyme, there's also sulfur. So keep in mind that sulfur is also an anomalous scatterer. So for both, this data set will be above the sulfur edge. So we are going to pick up the anomalous signal from sulfur. Okay. So on the right, that will be above the edge. You see that that's your model. And now if I display the anomalous difference maps, you see that all the sulfurs and both the chloride ion have been picked up in the map. And that's what we would expect if they are really chloride ion. If we now display the map below the absorption edge, now we don't pick up the chloride there. So by making the difference between this difference map or just looking at your maps, we can then identify if it's a chloride or not. So that's why it's quite important to select properly your wavelength or your energy for data collection, because that will basically allow you to check this anomalous difference map. So we did do that with quite a few different user samples. For example, that was a few years ago already. We did that with a ribosome, with the 78. This is really just to show you that we can do it with very large macromolecules. And as you can see, it diffracted only to 4 angstrom resolution, so not great. But for anomalous differences, it doesn't matter too much. And we did find potassium in quite a few places where previously magnesium was modeled, and that's because it's quite difficult to distinguish the two in your electron density maps. I should say nowadays, if someone works on the ribosome, that will be in cryo-EM, but cryo-EM doesn't tell you anything about there's no anomalous data in cryo-EM, so that's one of the benefits of crystallography, at least. Something a bit more subtle, we had some users that worked on therapeutic nucleic acids, and these are modified nucleic acids where basically one of the oxygen of a phosphate backbone has been replaced with a sulfur. That makes basically the molecule more stable and more potent. The thing is that when they synthesize this molecule, they don't know which stereoisomer binds to their protein, so either the S or the R. But because sulfur does have, we can collect anomalous data from sulfur and not from oxygen, we can distinguish that. And that's what you see here. So from our data, they could find out that basically the preference of binding of which stereoisomer compared to their protein. So. And for us, it's very trivial to do because I should say that compared to phasing, calculating anomalous difference maps is straightforward. Another example, again with sulfur. So if you haven't, you can see Izzy's poster below. So this was done also by Chris, who is now on I-23, and the group of EVOS. So the idea is that, as we all know, antibodies bind through their variable region to antigens. But what they show is that basically the hinge region right in the middle here is actually very important also for the agonist activities of the antibodies. And they show that basically the more rigidities, the more agonist activity it has for the IgG2 at least. So the issue is here, because it's a hinge, it's quite flexible, so the density is not great there. So they did quite a few mutations. So they needed to confirm the mutations, but also to make sure that the disulfide that you see here are really there, basically. And as I said, the electronicity map is not great in this region. So they did use instead anomalous difference map collected close to collected that wavelength where we can see the signal from sulfur. And you can see that they can nicely at least confirm which conformation they have in their crystals and relate this to the activity that they measured in vivo. And one last example. Yesterday we heard about XChem and the way that it's something that is still very strong in crystallography. And the idea is that you use these small fragments and then you start to build up from them to build your inhibitor, your molecule that will bind at the end to your protein. So this was done also with NSP1 from SARS-CoV-2. And that was initially published, but the user actually were not very confident about some of the fragments, like the one I'm showing here. So as you can see, they are quite flat, which means that you can basically fit them in two orientation quite often. So although the protein diffracted to quite high resolution, beta 1.4, because these bindings are not always great, they are flexible, or they don't have a high affinity because they are small, the density is not always very good. So what we did there is that we have taken data basically, I should say, so in green, that's the sulfur, and in pink, that's the chlorine. So we collected data to be able to assess if the chlorine and the sulfur were there. So looking at the first example, in magenta, that's the anomalous Fourier difference map, and you see that it fits quite well with the position of the sulfur. So that confirms their initial assessment. For the second fragment, we actually find that there are two peaks in the anomalous maps. So that means that this assignment is right, but actually you can also turn it 180 degrees to also have a sulfur here. So basically it's in a double conformation. And now this one is quite interesting because I would have fitted exactly this fragment in this way. At the top, it might be difficult to see, but there's a carbon, an amine, and below a methyl group. So quite strong in density. And here, just a chlorine. But actually, when you look at the anomalous difference maps, the peak is actually here, where you got the more density. And when you model, it's clear that it was built in the other way. So that's quite important because, as I said, you start with these molecules to build a larger molecule or to link them between them. So if you got the assignment wrong from the beginning, that's not a great start. So in conclusion, experimental data is still useful and necessary. I know that we are all aware of this here, but actually I think it's more for the biologists. And I think it's important that we tell them that we still do need experimental data. And you can get a lot of extra information if you collect anomalous data, and that will help you with your model building and in the confidence in your models. The nice thing with collecting anomalous data compared to F double prime or even compared to, for example, using like servers like Check My Method, things like that, is that you don't necessarily need a complete model or a completely refined model to calculate these maps. So so quite often we had, before AlphaFold, people coming with a polyanion model. They couldn't build the side chains. And that was enough basically to solve by molecular placement and to get the anomalous peaks. So that means we have pretty good confidence even if your model is not good. The other method do need you to have a pretty good model. Now, something to also bear in mind is that quite often it's pretty clear that there will be quite a few wrong assignments in the PDB, especially if you look at like sodium chloride that can look very similar to water. And the issue with that is that if we want to build the next machine learning algorithm that will be able to place properly metal ions or sodium chloride, we need to have very good data for that. AlphaFold worked because our data in the PDB for proteins was very good, but it's not the case for ions. So we do need to have a good database made of experimental data to be able to get this next machine learning algorithm to work. Okay, and I will stop here by thanking all the people in the I23 team, all our users and collaborators that provided the example that you've seen here, and also the organizer for inviting me to give a talk. Thank you for your attention.
A lovely talk. Oliver Smart, global phasing. Sorry, identify myself. Just to link, your last example is very interesting, but I do have a question just to link it to Elspeth's quote. Is if you've got a chlorine in your ligand, is it also worth just burning out and deliberately producing radiation damage? Do you think you'd get a signal that way?
By burning, you mean?
Yeah, when you have bromine in the
bromine, yes.
Bromine, it's routine. You get negative difference density, presumably because there's massive radiation damage. I mean, most PDB ligands with bromine have radiation. So, is it worthwhile thinking about doing that as well?
So we never tried that because we tend to collect very, very low dose and high multiplicity. But we can easily collect high dose and low multiplicity to test that. But we tend to be very, very careful with dose. So we never actually thought about. But yeah, that could be tried.
Could I make a comment on that? Elspeth Garman, Oxford. This is the method of radiation-induced phasing invented by Raymond Ravelli in the early 2000s, where you would do a very low dose data set, then you'd give it a burn because it was before the beams were intense enough. And then you'd do another data set where you'd blasted your sulfurs bonds to hell. And this, you had to have pretty well diffracting crystals. And we did do one unknown structure of fibronectin 2 modules with Raymond using this method. But then there was also a paper on doing it with bromine, which I can give you from about 2004, I think. I think it was Ramagozal. Does anyone remember? I've got it on my laptop. I'll give it to you. But yes, it's a way you make a hole basically, so it's sort of reverse MIR.
Actually, bromine will be very useful for nucleic acids also, because quite often you can synthesize nucleic acids with bromine for phasing. So
there's a question for you in Slack, but maybe you can answer that online because I think we're out of time.
Thank Kamel again.